Thank you. We're going to get started. So it's my pleasure to introduce Emily Darling. She is the speaker that we voted for to come speak to us. She's a marine biologist, and she's really made some great significant contributions to marine conservation. Her focus is on coral reef conservation in the face of environmental change. She completed her PhD at Simon Fraser University in 2012, and there she worked with Isabelle Cote, and she developed a method to predict how coral reefs respond to multiple stressors. And this dissertation earned her the Governor General Medal for the highest academic standing. And then for her postdoc, she received the very prestigious David Smith Conservation Research Fellowship, which she took to the University of North Carolina at Chapel Hill to work again on coral reef conservation. And now she's working with an NGO, the Wildlife Conservation Society, where she combines science with management to coordinate a global coral reef fisheries monitoring program. And then next year she's starting Banting Fellowship at University of Toronto. In addition to the conservation research she does, she has also done a lot of work with social media and how to use social media as a tool for scientists to communicate their research. And she also writes a lot of internet blogs to educate and stimulate interest in the public about conservation. She's been very productive in terms of her research throughout her career. Already has about 30 publications, and she only started her graduate work in 2006. And in addition to the awards I already mentioned, she also received the Glenn Gee Award for Marine Biology, the WWF Catherine Fuller Doctoral Fellowship, a Patty Foundation grant, various NSERC fellowships, and several others. She's apparently really good at giving talks, as she won 4 Best Talk Awards at various conferences, and she was selected as a plenary speaker at the International Marine Conservation Congress in 2014. So I think we'll really enjoy her talk today. So without further ado, please join me in welcoming Emily.
For the very gracious introduction. And thank you so much for all of you to coming, for Krista and Vincent for hosting my visit. It's been such a treat to spend so much time with so many different and interesting people this visit, so thank you so much. And of course the graduate students for inviting me. So, okay. How's the sound in the back? Good? Okay. So today I'd like to take you on a bit of a whirlwind through some ideas about how corals are moving in a stressed world, what we can learn about corals to other ecosystems, and how I think we can really start leveraging other sectors and other types of data like big data for conservation. I'd also just like to mention that it's a real honor to be able to talk about my research today for an hour, because I've had some really amazing conversations all day about NGOs and thinking, outside the ivory tower. So it's a really nice invitation to be able to talk about both things. So thank you. Before I start, this talk would not be possible without many of the people you see on the slide and more. And thank you, Julianne, for introducing some of those key people like Isabelle Cote, Tim McClanahan, and the folks I'll be working with at UNT starting. Some of the data at the end you're going to see are unpublished data from a number of scientists throughout the Indian and Pacific Oceans, and I just wanted to recognize that contribution here first. So I'm just going to dive right in. That was a pun as a marine biologist. But it's been a really weird few weeks to be a coral reef scientist. These are pictures hot off the press, hot off social media, of what the world's most iconic coral reef, the Great Barrier Reef, looks like today. 1,000 kilometers of the Great Barrier Reef has bleached. This is a stress response to superheated waters. 50% of the Great Barrier Reef, its northern section, has seen mortality. That's dead coral. That will likely take decades to recover. We are seeing this bleaching at some of the world's most pristine sites, not just the Great Barrier Reef, and also at very deep sites. This is a photo from 18 meters depth, typically what we might think to be a depth refuge of cooler waters for coral. We're still seeing bleaching. This is timely news, this event. This is the headline of The Guardian, mass coral bleaching now affecting half of Australia's Great Barrier Reef. This is from over the weekend. The New York Times, climate-related death of corals around the world alarms scientists. I agree, I am alarmed. The Washington Post talks about why dead coral reefs could mark the beginning of dangerous climate change for coral reefs, but also for the rest of our ecosystems and our societies. So things are happening, and it's a really timely time to give this talk. So what I'd like to dive into here is, well, how did we get here? And where do we go from here? And how does conservation science help inform our decisions to really try to affect change in an ecosystem that's really changing before our eyes? So today I'd like to take you on a tour of some of my research, and I'd like to propose 4 things that ecology can contribute to conservation. First, I'd like to show you that interactions among multiple stressors are important, and there's something that ecological theory can help us understand. I'd like to talk about how species traits and life histories can also help us predict how communities disassemble and perhaps reassemble to global change. I'm going to touch on why species matter for when we think about managing resilience to climate change in particular. And finally, I'd like to focus on that big data portion of it, which is, I think we can use big data to find some solutions and think about climate refuges as priorities for conservation. So before I dive in, I thought I'd just give you what my definition of conservation is from a Google search. But I think we need to really make the distinction between conservation and conservation science. Conservation is defined as an ethic of resource use, allocation, and protection. And I'd really like to highlight the role of conservation science, which is the scientific discipline that seeks to understand the health of the natural world, ecosystems, biological diversity, but also to understand how we can sustainably use these resources. So I am a conservation scientist, and I thought I'd just give you a quick overview of how I ended up here. So I am currently a scientist, a staff scientist at a nonprofit NGO. WCS, which you might not know, has over 500 conservation programs around the world. We work in 60 countries, and we have over 200 PhD scientists on staff. So there are jobs when you finish grad school, I promise. But to get here, I took quite an academic path, starting with a postdoc and SFU and Bachelor's of Queens, which was so nicely introduced earlier. So you can also take a path through Canadian universities and end up working around the world. And I'll also be going back into the Canadian university system for a couple of years just down the train ride, so happy to really make connections with folks during this visit. So diving in, why should we care about tropical coral reefs bleaching? Well, they're incredibly biodiverse and species-rich ecosystems. They support 1/3 of ocean biodiversity. These species-rich systems have a rich scientific history. O'Connell's intermediate disturbance hypothesis was born from coral reefs and tropical rainforests. Darwin's first monograph was titled Coral Reefs on his Eagle adventure. And perhaps more practically these days, 500 million people around the world depend on coral reefs for either their livelihoods, shelter from storms and tsunamis, or cultural practices as well. So there's a lot of ways we can dig into coral reefs and conservation. The story I'd like to tell you about coral reefs starts on the other side of the world in Kenya, where I lived for 6 years on and off during my PhD. During these 6 years, I was working with the Wildlife Conservation Society as a graduate student at SFU, and ended up surveying reefs about every 15 kilometers between Malindi and Kasiti. And most importantly, ended up working with an extraordinary group of scientists, interdisciplinary scientists, social scientists, you see here, as well as local fishermen, who I learned a lot from. Small-scale fisheries in Kenya, where our story starts, look very different from fisheries we're used to here in Canada. These are low-tech fisheries where there's a lot of effort, so there's a lot of fishermen out on the reefs, either walking out like this guy here, or out in boats with no motors, with no sails, just trying to scare fish into a net, sometimes by slapping the water with their hands and trying to get fish into a net. When they land those fish, they'll sell them to put money in their pockets, or they'll bring them back to their families to have food on the table. This is a notoriously challenging fishery to manage because it's multi-gear and multi-species, like so many fisheries in developing countries. And because of that high effort and notorious difficulty to manage, you can see the impacts of these fishing activities on the ecosystem. Here you can see fishermen pulling fish, a whole bunch of different types of fish out of the water that can have indirect effects on the foundation species, the corals, which I'll mostly talk about today. But you can also have direct impacts. So here fishermen are stepping on corals. These are very shallow places, so as dugout canoes are pulled in or out at low tide, you can have breaking and damaging corals. And that's a problem because they provide the underwater architecture of reefs. So when I first started diving underwater on reefs, in Kenya, the fish reefs looked a lot like this. You don't see many fish. The ones you do are kind of scared looking and hiding at the back of the photo. But you do see corals. You just see a few different species. These are called massive Paredes corals or mushroom corals. And there's a lot of dead substrates, some algae, but there's some corals. You'd also see a lot of sea urchins because of a trophic cascade from the removal of predators. But not all of Kenya's reefs are fished, and in fact, just like Kenya's famous national parks on land, you also have very effective and enforced national parks in the ocean. When you dive on parks like this, no-take marine reserves, there's been a long history of work from WCS scientists showing that these parks have up to 10 times more fish biomass than on the fish reefs. So these parks work, they're not paper parks. But as a coral, or someone getting a wannabe coral ecologist, I wanted to look past the fish and actually look at what the corals were doing there instead. And when you swim up and look at the corals, they're actually the same species as what you saw on those fish reefs. There's just maybe a bit more of them. These are, again, more of these mushroom, massive Paredes corals. So why is this? Well, this is actually a legacy of climate change. In 1998, a lot of these reefs might have looked like this. In 1998, there was a very strong El Nino event, similar to the one we've been seeing over the last 18 months, build, and it led to the warmest recorded water temperatures in Kenya on history, or recorded in history. So this led corals to bleach, which is a stress response that can lead to mass coral mortality, and this has really been the elephant in the room for how we manage and conserve coral reefs. We typically think about managing local stressors, but what do we do when we have this global stressor that we can't actually manage for? Could this be responsible for why corals looked different, or didn't actually look so different? So I'd like to show you first some long-term data sets of change collected by WCS scientists. And what we did was we looked at live coral covers. This is the amount of living coral on a reef over time. And I just want you to remember 1998 and think of that as having some kind of gloomy shadow, unfortunately, about what's going on today. So 1998 is a lot like what's happening right now. So 1998, what happened? Well, before bleaching, marine reserves, or those marine parks, I might go back and forth in how I describe those, they had about twice as much living coral cover as fish reefs, which isn't too surprising. 1998 hit, and the narrative in the conservation literature back then might have been, well, the reefs that are more locally protected, they should be more resilient to climate change. So that's what we expected, and how we wanted to start digging into this idea of multiple stressors. Fish reefs, they're more stressed, they're probably going to be more stressed when they have bleaching as well. So what did we find? Well, we actually found the opposite. Reserves lost more coral, and they lost it faster than the fish reefs. After this bleaching event, both reefs recovered at approximately the same rate up until 2007. If we extended this data set until now, we would probably see the marine reserves starting to pull ahead of the fish reefs. But it's still a little bit sobering that, over this chunk of time, really a decade, we're not seeing differences in recovery rates. We wanted to dig into multiple stressors and tease this apart. And so what we did was use a meta-analysis approach of effect sizes and say, well, what's the effect of fishing? And so what we did was we compared the parks, the fish reefs, before bleaching. And then we said, well, what's the effect of bleaching? Well, we compared just the parks before and after bleaching. So using long-term monitoring data sets to try to tease apart these stressors. And then we said, well, what happens if we add these up into a null model of interaction effects? And then what did we actually see in terms of what happened on the fish reefs after bleaching versus the parks before bleaching? This is where it gets interesting because we found, first of all, that bleaching had a higher effect size on coral loss than fishing. So climate change here was the more dominant driver of the changes we saw. And when we added those up into a null model, which is a way we typically think about interactions, and compare that to what we saw, we didn't see a synergistic effect. Instead, we saw more of an additive or antagonistic effect. I'm not going to stop now and sort of explain those different terms, but I'm happy to chat about that after. But just wanted to highlight that this was surprising. because a lot of the literature that I'm sure all of you are reading are talking about synergies. And so why are we not seeing a synergy? Well, we also dug into that in another meta-analysis way, just stepping aside from coral reefs. And so what is actually the evidence for ecological synergies? In our meta-analysis approach, we found that, well, actually there's not a ton of evidence. And in fact, of over 100 experiments, we found about a third synergies, 1/3 additive effects, and 1/3 antagonisms. So that starts to suggest that if you're really looking at the impacts of multiple stressors, you can't just assume it's a synergy. We really need to disentangle the mechanisms of what's happening and think about how that applies to conservation. I just wanted to quickly highlight some of our work on that just came out this year, thinking about these interactions and their importance for conservation. The one things I wanted to highlight was all of these interaction types are increasing in how we're talking about them in the literature, but we are talking about synergies more. That's the red line. We're also becoming a lot more better at jargon around multiple stressors, and this is not something that's helpful for conservation. Here we synthesize a number of recent studies that propose what happens if you have stressors in the same direction or different directions. You can see some of the red and blue arrows don't quite overlap, so the literature is not agreeing really on what is a synergy and antagonism. We're trying to define things like super synergy, and this jargon isn't terribly helpful, we argue. But you can start to look at different levels of ecological organization and say which ones are we more likely to see different types of effect. And the only thing I wanted to highlight here is that if we look at synergies, we seem to see some evidence in a meta-analysis of meta-analyses. We called it a meta-meta-analysis. We're pretty proud of ourselves. We seem to see some evidence for synergies coming more out of physiological or individual level studies, whereas individual population, community, ecosystem levels seem to have some evidence that those associate more with antagonisms. So going back into corals, that perhaps is not as surprising as what we saw. So going back to corals on our whirlwind tour, why wasn't this a synergy? And what can we learn about the fact that we didn't see a synergy? Well, we thought that there might be something happening in the coral assemblages, the types of corals themselves, to help us understand why marine reserves lost so much coral. Because we know that coral communities are changing around the world. Over the last few decades, in both the Caribbean and Indo-Pacific oceans, we're seeing this loss of large branching and plating structural corals. And they're what's being left in the community, or their replacement, with more weedy species. This is an Agaricea coral. It's called a potato chip coral because as soon as you kind of look at it funny, it just crumbles. Below are those mushroom parietes massive corals. They typically live for a long time. They're hardier. They can kind of, these are the corals that can be 800 years old. As big, huge corals. People dive down in like tourism photos. There's this evidence in the literature that we're seeing this replacement of coral communities, but there's actually no global quantitative framework to be able to test those ideas or apply them to our Kenya data set. So in what seemed like a good idea at the time, as I'm sure many of you are familiar with, we decided to sit down, or I decided to sit down in front of taxonomic monographs, papers, Web of Science never spend too much time together, and the literature to say, well, how can we quantify coral life history strategies? And I brought together every bit of information I could for every coral species in the world on 11 major traits that we think talk about how corals make a living underwater. So what type of colonies they produce, how fast do they grow, their maximum size, sort of classic life history traits that we think of in other ecological theory. I was able to collect bits of information for about 800 species, which isn't bad since we estimate there's about 1,000 species of reef corals out there. And this came from over 300 of those references. At the end of the day, we had enough trait information on about 143 species from over 250 references that will form the analysis I'm going to show you in the next slide. And I just wanted to highlight there's a lot more information on this. Actually, all of the trait information is now freely available online at this really awesome website, coraltraits.org. So when we use a hierarchical approach, and we use non-parametric multivariate ANOVAs to say how many different clusters do we have, we use random forest classification trees to say what are actually driving these different groups, we come up with evidence for 4 groups that the traits group species into. And when we look at the traits underlying each of those groups, we can put some labels on them. And here's what those labels mean. We seem to see some evidence for what we're calling a competitively dominant or a competitively dominant life history. These are those branching and plating kind of foundation species corals. They have fast growth rates, they can have larger colony sizes. We're also seeing collections of traits that we think are associated with more of a stress-tolerant life history. So those are those massive or encrusting boulder corals. They grow slowly, they're long-lived, they can have high fecundity when they want to, so when the time is right, they can pump out a lot of eggs. And we also see some evidence for perhaps a weedy life history, which is predominantly defined by having a brooding coral life history, which means that the fertilization happens sort of inside, or the coral is inside and spit out right next to it. And we think that that can help corals take over space opportunistically after a disturbance. So a couple different types of life histories here, and this was interesting because many of the plant ecologists in the room will start seeing, signs of Grimes' triangle for corals. Does this apply? Here is where we really just drew a triangle. I drew a triangle around the plots. Maybe, maybe not. There's certainly some interesting arguments to be for or against. Another interesting thing is, well, what about all this missing space? I think if we start thinking about life history traits of algae or sponges, we can start thinking about Grimes' triangle of benthic communities on reefs, which is more similar to plants in general. But anyways, some interesting insights are certainly there to be made between plants and corals as sessile organisms, often clonal, that need to make their way in an increasingly hostile world. I just wanted to highlight some recent work as well on this topic. As I mentioned, all of this data is now available online as a kind of global collaboration of coral trait information led by Josh Maiden at Macquarie University. Just an amazing effort. We also have a paper out about a trait-based approach to advance coral reef science. So for folks in the room who are looking for data and questions, I would recommend these two papers, as that's sort of our current thinking around traits and corals. So coming back to Kenya, now that we have those life histories, do these life histories actually explain community disassembly to these multiple stressors that I had familiarized you with? So I'd like to show you again this time series. We've extended a little bit. We're going to go 1991 to 2011. Don't forget the importance of that 1998 event. And what I've done is I'm going to pull out the marine reserves and show you those first. And I'm just going to focus on the three primary life history strategies of corals. So before bleaching, marine reserves had fairly functionally intact assemblages where we had living coral and all living life histories. Bleaching happened. And not surprisingly to the ecologists in the room, some life histories were more sensitive to that shock than others. The competitively dominant species found themselves in very unideal conditions and promptly died. The weedy corals equally promptly died. And the only things that hung on in these communities were these stress-tolerant corals that sort of hunkered down and was like, no problem, I lived for 800 years, this is no big deal. But what we really saw was this real reorganization of the coral community that seemed consistent with those previously reported community trends from around the world. If we look at fish reefs, something really else happened that was different. Before 1998, the fish reefs looked very different from the marine reserves. There were no competitively dominant species, likely because of those indirect and direct impacts of fishing on the corals. And instead we had a community of fairly weedy and stress-tolerant corals. Bleaching happened, and here was where we were really interested in what was going to happen to these already stressed reefs. Bleaching hit, we were prepared for the worst, and actually nothing really much happened at all. There were some differences, the relative dominance of red and blue, blue and green kind of switched before and after. But if you take a step back, this assemblage of more weedy and stress-tolerant corals just sort of kept pushing through this big event and sort of hunkered down and stayed there. So very different community trends, and certainly some inference that we're able to make from these life histories. Okay, well, so what? If you're a reef fish, or if you're a non-coral or reef fish biologist or another biologist in the room, you might be thinking, so what? I'd like to argue that this story we saw on Kenyan coral reefs actually critically affects how we think about conservation and management in the face of climate change. So this is the way we typically think about managing for climate change on coral reefs, and I would argue a lot of ecosystems around the world. We expect that ecosystems in a more pristine state can take some amount of climate stress. So as climate stress moves from low to high, so the ecosystems can sort of keep up until they reach some kind of tipping point. And this is certainly if the ecosystem exhibits nonlinear dynamics, we see tipping points. For corals, that tipping point looks like bleaching, and they typically then fall off a cliff into a very poor or degraded state quite quickly when you have this mass mortality. The way we've approached resilience and sort of bleaching is that as ecosystem states are degraded through things like fishing or disease or pollution, that we're pushing down the ecosystem state to make it more poor, and we're actually shifting that tipping point to climate change to the left, so that more stressed systems can only absorb so much stress before they tip. I'm seeing nodding in the audience, excellent. And so saying, you can do that again, this tipping point keeps shifting to the left, and so what underpins our management is that if we just push systems back to a pristine state, we'll also be pushing their tipping point back to the right, and this is resilience, where the capacity of a system to absorb and recover from disturbance. So tipping points in ecosystem states in the literature, this has been the conventional wisdom. But what I'd like to say that we learned from Kenya is actually that species and traits matter. And what we found was that these competitive corals, these ones that were those really important framework builders, they were actually disproportionately vulnerable to bleaching. And instead what we saw was that stress-tolerant and weedy corals were less impacted by bleaching. And that's likely due to a co-tolerance between these two different types of stressors. So that things that are disproportionately affected by fishing were also the ones, sorry, I'm gonna get that wrong. I'm gonna time out that one, but this does have to do with co-tolerance. So what this means then is that we might actually be seeing an alternative view of resilience, whereby when we have coral cover at a high state or this more pristine ecosystem state, they might actually have a fairly low tipping point to climate stress. So here, competitive corals that are sensitive to climate change, they seem to tip into this coral poor state much quickly. And in fact, as we saw more degraded or stressed systems, that tipping point actually shifted in the other direction to the right. So that more degraded systems, those fish reefs, were actually able to take more climate stress because they're already composed of stress-tolerant and weedy corals. So our differential susceptibilities of species to stress, and particularly those correlated tolerances, perhaps presents an alternative view. And the interesting part about this alternative view is that as you manage your corals or your systems back to a more pristine state, you may be actually inherently increasing their vulnerability to climate change. Now again, we're focusing mostly on the resistance aspect of resilience, and resilience is both resistance and recovery. So here we're focusing on just one part of that. I'm certainly not suggesting that we need to fish all reefs around the world to increase their resistance to climate change. That would obviously be incredibly short-sighted. But I do just want to highlight that, by thinking about ecological theory and merging that with natural history observations, long-term data sets, you can actually start to think about some alternative views to the world, which have very important consequences for how we manage ecosystems in the face of climate change. So this perhaps means that future coral reefs, as they keep getting hit by multiple stressors, they might be flatter and less diverse. But they might be able to bounce along the bottom of the barrel for longer than we're perhaps expecting. Does this really mean that pristine reefs are more sensitive to bleaching? Well, that's certainly the evidence that we're seeing in the Great Barrier Reef right now, as well as in the Central Pacific. This is a tweet from the University of Victoria. Even pristine sites in Christmas Island, now have 75% coral mortality and 10% coral bleaching. So that's sort of 10% remaining bleaching. That 75% mortality was from this bleaching event. Australia's Great Barrier Reef hit by worst bleaching ever recorded. The quote at the bottom is from Professor Terry Hughes, who said, "This has been the saddest research trip of my life." The Great Barrier Reef and Central Pacific, these are pristine coral reefs. These are some of the world's best managed coral reefs. And it's certainly suggesting that simply having a park does not protect something from bleaching. Being pristine or remote does not protect something from bleaching. And that's not surprising because our entire oceans are heating up. Our oceans are also acidifying. Climate change is being forced at a scale much larger than our local actions. So what do we do, and how do I not end you on a doom and gloom note? Well, actually, I'm only about halfway through, so it's going to pick up, I promise. But what I want to show you here is that the ocean is a weird and wonderful place. The ocean is not homogeneous. The ocean has currents and channels and depth. The ocean, has these very deep canyons that pull cool water up. You can see gyres and eddies in these massive ocean systems. And I just wanted to highlight that Kenya, the one place I've been telling you about so far, is here where this red arrow is. And it's typically a place where we see a lot of this hot water pooling and sitting and being forced up from the Indian Ocean, which is why we saw such strong effects of that 1998 bleaching. And indeed, we're also seeing bleaching along that coastline right now. But as a WCS scientist and working with WCS, I've also had the incredible privilege of seeing other coral reefs in this region. And I'd like to show you some photos from 2 expeditions we've done to northwest Madagascar and to Mayotte, which is in this northern Mozambique Channel ecoregion that is structured by these deep canyons and eddies and gyres. So we have a lot more coral mixing, and do we see something different happening there than we did in Kenya? And I would like to say that yes, we do. So on these research trips, this is what corals look like today. They did bleach in 1998, and they're looking very nice today. Here is very high coral cover of these branching and plating corals, those competitive dominants that we know are quite sensitive. This is another photo of just incredibly high coral cover in Madagascar. Is this a climate refuge? Is this a place that had a more stable environment, or perhaps a more variable environment? But does it have some properties that allow these corals to either escape the worst impacts of climate change or have stronger recovery coming back from climate change? And is this a hotspot of diversity? Is this something that we can leverage when we think about how coral reefs or how other ecosystems are going to survive climate change? Refuges are important for conservation because they suggest it might not all be doom and gloom. The world's leading scientists are finding, evidence for adaptation and survival. Dr. Andrew Baker says we're starting to see corals resisting heat stress in a variety of different ways. Steve Palumbi says corals are getting mad. They can move, acclimate, adapt, or die. So they do have more options from just dying. That's good. And so these things are possible, and how could refuges be a key for that? Well, if we take that the ocean is this weird and wonderful place, and there's a lot of different things happening, what happens if you're a coral kind of stuck in the middle, if you're in one of these superheated waters? Well, one thing you could do is that over multiple generations, you could hop-skip your way down to cooler waters. This is consistent with sort of poleward migration towards cooler waters. This wouldn't be the adult coral themselves, because obviously they're sessile, but it would be through subsequent generations of larval dispersal. But when they get down to, say, the tip of South Africa, where it is actually quite cold, I can attest for that underwater, they still have limitations today. It might be too cold still. And these reefs are typically covered in more macroalgae, or they're not quite the right settlement habitat for baby corals. There's also a really interesting paper published in Science last year that said they don't have enough winter light. So while climate change is warming the oceans and changing temperature, it's not changing the rotation of the Earth, so that light will change. Although I think there was just a story about melting glaciers and the Earth's tilt is tipping, so we might have to revisit that. But there might be some limitations to high latitude